So what I will tell you about today is our work on uncovering transcription condensate organization with single molecule resolution. I will spend a little bit of time introducing the system, going over some of the work that really set the stage for this, and then tell you more about our mostly super resolution work in fixed cells, but also take a little bit of time to start going into live cell single particle tracking work to really understand what these transcription condensates are doing, how they function in a very physical sense. We're located in the physics department. All right, here we go. All right, so on this title slide, you see a colony of mouse embryonic stem cells, and we stained RNA polymerase II in these cells, the then super resolution on them, and you can see these dense accumulation. Let me just see if I can find my laser pointer over here. There we go. You can see these dense accumulations, PO2 condensates in essentially all of these stem cell nuclei. So all of this happens on top of densely packed chromatin throughout the entire cell nucleus. We have our 2 meters or 6 feet of DNA that we need to compact into a nucleus that is maybe 10 micrometers in diameter on average. So that's 4 gigabytes of information in human cells that is very densely compacted in here. So somewhere in this spaghetti bowl, if you will, of DNA fiber, we have located genes, histones, wrapping proteins help with organizing the DNA to make functional regions accessible and allow us to use in each specific cell type the parts of the genetic information that we need. All right, how does transcription work? Here's my very simple schematic, just to give a little bit of an intro to that. Somewhere in this DNA, we have genes located, some 20,000 or so in the human genome. Only about a third of them are actually used in any given cell type, and they are demarcated by transcription sites that are flanked by promoter regions that will bind regulatory proteins, transcription factors, co-activators, and so on, that help recruit RNA polymerase II to these promoters and then initiate transcription. But we also need more distal enhancer regions that also attract transcription factors, co-activators, and so on, and that somehow communicate information to the promoters to actually facilitate transcription from the target promoters. One protein complex that is very important for that is the mediator complex that is thought to bridge this contact between enhancer and promoter. That's where it gets its name from. Mediator mediates the contact, the communication between enhancers and promoters. And if all of this finally comes together, we can get the polymerase loaded onto the gene and start producing transcripts from that target gene. So it's becoming increasingly clear that these enhancers are really the important regions that control which of these cell type specific genes become active, which subset of the 20,000, more specifically, in the first instance, which transcription factors become active to then define the state of the cell. Is it a fat cell, a bone cell, stem cell, skin cell, and so on. How do you identify enhancers? That's often done with ChIP-seq data, where you pull down a certain protein mark using antibodies, then sequence the DNA that comes down with them and generate this read density map that indicates where you find typical enhancer marks, like these transcription factors, PO2 mediator, or certain histone marks. And so we would have our target gene over here in blue, and in red, different signature where we find an enhancer. So it's characterized, for example, in this little subset over here with this peak of transcription factors, and there's some mediator present there as well. Now, it turns out that some enhancers, specifically mostly those that control master transcription factors that drive the cell type identification, often have collections of these single enhancer peaks. Sometimes you call them clustered enhancers or super enhancers. So much larger integrated control regions for these master transcription factor genes that have the same kind of marks that our typical enhancers would have, but in much larger quantity and often in these little clustered elements. And if you rank enhancers by the mediator signal, and this is data from Rick Young's lab at the Whitehead Institute, then you will find that some of these top hits, the strongest enhancers by mediator signal in ChIP-seq, are exactly those cell type specific master transcription factors in our mouse embryonic stem cells, for example, SOX2, ESRB, KL4, NANOG, OC4, you name it. They're all appearing at the very far end, the very far right of this rank ordered plot of mediator signal at enhancers. So that's all nice and well, but it raises a question. Right? So if we have this extended stretch of a clustered enhancer or a super enhancer, and that collectively somehow controls a target promoter, then we have to find a mechanism for all of these different elements that can be stretched out over tens of kilobases to simultaneously and collectively regulate expression of the promoter. Sometimes these elements are merely redundant. They give some robustness to the signal, but sometimes you get examples where multiple enhancers at the same time talk to the same promoter. And what's more, these enhancers are not exclusive to a single promoter. Some of them are able to control different promoters in the same neighborhood as well. So that is at odds with this simple one-to-one contact model of enhancer-promoter communication that I presented initially. So how can we make that work? One idea that has garnered a lot of attention in recent years is the idea that maybe we could form different distinct microenvironments through phase separation in the cell nucleus. We could form biomolecular condensates that concentrate transcription machinery at these super enhancers and collectively provide all of these different factors that are needed for transcription activation. The condensates obviously are found these days in very many biochemical processes in the cell, in the nucleus, outside of the nucleus. So there are a number of great reviews that discuss all the basics of condensate biology and present you whole lists of different processes, biological processes that condensates are now implicated in. We think that they might also play a role in transcription regulation. And why is that? How would that work? Well, the idea goes back to the fact that many of these transcription factors, co-activators, are globular proteins that also harbor disordered protein domains. Not the entire protein is disordered, but certain domains of these proteins are disordered.